Hello everybody, my name is Aaron Hilliard. Welcome to Mushroom Wonderland. On this video, I'm going to show you how to identify a mushroom that you find in the wild. Maybe you're just walking down the trail and you see a mushroom growing there and you want to know how to identify it. This is the video for you. So thanks for joining Mushroom Wonderland. Make sure to hit subscribe and come with me as we discover how to identify a wild mushroom.
Mushroom Wonderland.
So I make a lot of videos on this channel all about wild mushrooms, but it was brought to my attention that I don't really have a video that shows you just how to identify a random mushroom in the wild. And so yesterday I had the pleasure of going into the woods, and one of the first mushrooms that I encountered was unusual to me. It was a mushroom that I was unfamiliar with. So once you find a mushroom like this and you start to look at it, even for somebody who's experienced, I might be going, I don't know what this is. I've never seen this mushroom before. Where do I start? What do I start doing to identify this mushroom? First, let's go into the woods and see these mushrooms that I discovered growing on the side of the trail. A beautiful little flush of mushrooms that I found to be unfamiliar. And we're gonna have a look at these and then learn how to identify them. So I was just walking down the trail and I came across these three mushrooms, pretty handsome, medium-sized mushrooms growing on the side of the trail, coming out of the moss. So one of the first things that we need to do in order to identify mushrooms is to understand the parts of a mushroom. So these mushrooms are pretty mushroomy in shape. And I say that because there's a lot of different shapes of mushrooms. Some of them grow on trees, some of them look like little orange boogers, some of them look like the Mario mushroom. And so this mushroom looks a little bit more like a typical mushroom, meaning that it has a cap and a stem and it has gills underneath. So let's look at these parts a little bit more in depth. So the whole mushroom here, this is called a fruiting body or a basidiocarp if you want to get really scientific about it. But it consists of a cap and then a stem, but in fungal terms we call that a stipe. So the stipe is going to be the stem portion of the fruiting body, and then the cap is the wide part, and then we have something called lamellae, which is the scientific name for gills. So if you're familiar with any kind of mushrooms like the ones that you get in the grocery store, like portobello mushrooms, they have gills underneath. And so that's one thing that I notice about these mushrooms right away, is that they have a stipe and a cap with gills on them. So in order to properly identify a mushroom, you have to pluck it from the soil and look at the underside of it, and you have to make sure to excavate the entire base of the mushroom so that you can see the whole thing. Sometimes key identifying features are going to be found at the very bottom of the stem where it attaches to the substrate that it's growing from. And so when I say substrate, I mean either soil or wood typically are the two kinds of substrates that we find mushroom fruiting bodies growing from. So as I pluck one of these mushrooms gently and carefully out of the soil, I notice that it has these gills underneath. And when I take a really close look at these gills, I'm going to realize that they kind of run down the stem just a little bit, and that is called decurrent. So a lot of mushrooms share this feature where the gills can sometimes run down the stipe. And in fact, a lot of mushrooms share this characteristic, and so that is one of the key identifying features. All of these little things put together can really lead us to a positive identification of a mushroom. We look at the size of the mushroom. These mushrooms are about 3.5 to 4 inches tall from the bottom of the stipe to the top of the cap. I'm starting to identify just the basic features of the mushroom, the cap, the gills, and the stipe. One of the main ways to identify a mushroom is going to be through spore color. So spores are like seeds of a mushroom. And a mushroom, we call that a fruiting body because a fruit grows some seeds in it, and once they become mature, it'll fall off of the plant, and then those seeds can grow from within the pulp of that fruit. But a fruiting body, like a mushroom, actually grows this structure that is more of a factory, and it's producing millions and millions of what we call spores. And so these are microscopic cells that will germinate and start a new fungal colony if they land in the right place. And so these fruiting bodies are producing spores by the millions, and they're dropping spores into small air currents and wind. They're getting caught on the hair of little animals. They're getting caught on my pant legs. And when you pluck a mushroom, you're actually putting a lot of spores into the environment. So not only is it necessary to pick mushrooms to be able to identify them, but it doesn't hurt them. So I want to make that clear. For identification purposes, you've got to be able to pluck the whole mushroom from the base and everything. And by lifting that mushroom up into the air, you're actually allowing for more spores to go flying off into the air, catch some wind currents, and get out there into the forest deeper. Whereas when it's only 3 inches off the ground, not a lot of those spores are going to make it elsewhere. So in a way, you're actually doing a favor to the fungus by plucking this fruiting body. So next, I want to get some very nice photos in situ. That means in the situation. That means where they're growing in the wild. So I'm going to stage these mushrooms in a way so that a photograph will kind of get all of the different features all in one picture. So if there's really young ones, I want to include those in the photo, all the way up to big mature ones, because I want to document exactly what these look like the moment that they were plucked or that they're living, because they start to deteriorate really fast after they're picked. I'm going to take some photos, some really nice close-ups with the mushroom in my hand, and I'm going to go immediately to an app on my smartphone called iNaturalist. And I'm not getting paid by iNaturalist to say this. This is just kind of like what the modern citizen science community is doing to help identify mushrooms and to start to get a database going of what mushrooms grow where. So I'm going to take a bunch of really nice quality photos with my cell phone or a fancy camera if I have one of those, but a cell phone can work. I suggest putting it on a small tripod and setting the timer so that it's not vibrating, so that it is as clear of a picture as you can get. And I'm going to take several photos from several angles, and I'm going to upload them onto iNaturalist. This video really isn't about iNaturalist, but I just got to say that if you're really getting involved in mycology, that you've got to be on iNaturalist. Also, a website called Mushroom Observer. These are both great places to upload photos of your mushroom finds so that other people can see them. You might get help with identification from other people online, and then it can help science in the future. If somebody wants to find this particular mushroom, or if they want to know its range, they can see that, yes, here's these photos. It's got a geolocation, exactly where it was found. We know that this mushroom does occur in this particular forest. And if the same mushroom is occurring in other forests, it's just nice to know with the range of these fruiting bodies, where they're popping up. So after I take my nice photos for iNaturalist, I'm going to collect a specimen to take home. Some people will carry a basket into the woods. Oftentimes I'll carry a small little fishing tackle box that I could carry several different specimens and just throw it in my backpack. And this is a perfect example of why, because I'm unfamiliar with this mushroom. Just by looking at the gills, I can't really tell what color spores are coming out of this mushroom. So spore color is probably the biggest factor when learning to identify a mushroom. So I'm going to collect this mushroom, I'm going to get it home, and I'm going to take a spore print. All right, so what I'm doing for this particular mushroom is I'm going to take a spore print. So I just have this white envelope, a return envelope that I don't need, and I'm going to reuse it and to make a spore print. I have a pretty good feeling that this is going to come back pink, but I'm also doing it over a microscope slide, and I can analyze the spore size in the microscope using a computer program called Pixiemeter, and we'll count about 30 spores and get an average size on them, and then I can really compare them to some literature about mushrooms in this pink spored genus. I'm assuming it's a pink spore. I could be wrong. It's kind of fun when you find a mushroom that you're not 100% sure about to do this kind of research. You take it home, you take a spore print. When we get that spore print, we'll go ahead and do the rest of the work, and it just makes mycology way more fun when you can go down the rabbit hole like this and really get into identification of different species that maybe you don't see all the time, and maybe you have a little tiny breakthrough in citizen science. You know, maybe that mushroom has never been discovered near you, so it's kind of exciting that way, and I like that. So that's why I do this. All right, it's been a full 24 hours. Let's take a look at this spore print I'm gonna lift the cup off of there. And here's our cap. Oh, look at that. So it might not be that obvious to you, but this is a pink spore print. And I can see that pinkness in the gills. Now we can start the process of looking through a dichotomous key of a mushroom guide that has a key in it. All right, so now we're gonna look at what's called a dichotomous key in this, the famed Mushroom Bible, Mushrooms Demystified by David Aurora. It's kind of an older book, but it really covers a lot of mushrooms, and it has great keys in it. So a dichotomous key, the word dichotomous meaning two. So you have two questions. You answer one of the questions, it'll lead you to the next question. So right here, key to the major groups of fleshy fungi, page 52. So here we go. Here's some options to look at. What kind of mushroom do we have? So this one, a fruiting body with cap and stalk, or just a cap. Spores borne on gills. So gilled mushrooms. Here's like chanterelle type mushrooms, boletes, polypores. We're going to start here, page 58. Agarics, or gilled mushrooms. So key to the agaricales. So these are all mushrooms with gills. And then here's your dichotomous key. See, there's two options for number one, two options for number two, three, and so on. So we're going to start at one. Gills deformed or contorted or branched to form large cavities. No. Nod as above. Spores forcibly discharged, hence a spore print obtainable. Yeah, we'll go to that. Two. Spore print white to buff. Nope. Yellow to orange, lilac tinged. Nope. Spore print some other color. Pinkish, salmon. Yep, that would be us. So we're going to skip all the way to 10. See, it says 10 right there. We'll come all the way down to 10. Spore print pinkish to flesh-colored salmon, pinkish cinnamon, or sorted reddish. Yep, 11. So we'll go to 11. Universal veil present. No, that's not right. And if you know your mushroom parts, you know the veil is going to be a ring or a skirt on the stem. So not as above. Vulva typically absent. Yep. So we're going to move to 12. Gills typically free at maturity. They're not free. These gills are decurrent. So 12, not as above. Let's go to 13. Odor strong. Did not have a strong odor. So not as above. 14. Spore print pale pinkish, pinkish buff, pinkish cream, gills sometimes purple tinged. Spore print deeper in color, sorted reddish, salmon, gills often not purple. Yeah, let's go to 15. That sounds right. And so the one thing with this is you got to be pretty accurate or it could mislead you. And if you get misled at one point during the key because you didn't describe it correctly, then it could send you somewhere that you don't want to go. Usually growing in clusters. Flesh fragile. Nope, that's not us. Not as above. Spores typically angular in the end or side view, which I do know that they are angular thanks to microscopy. So Entomomiaceae, page 238. So that leads us right to Entoloma. The family of Entoloma is Entomomiaceae. Right here. So now it leads us to another key. So one, growing on dung or other mushrooms. Nope. Growing on wood, humus, or ground. Yeah. Not humus, but humus. So we're going to go to 2. Stalk lateral to absent, fruiting body small, usually on wood. That's not it. Stalk present, usually central or slightly off center, on ground, sometimes on wood. Yes. So 7. We're going to jump down to 7. Gills purple or purple tinged. Spores not angular. That's not right. Spores not with above feature. Gills rarely purple. If so, then spores angular. Yep, let's go to 8. Gills tinged yellow. Nope. So 9, not as above. Widely distributed. E, rhodopolium group, and others below. So let's go right to here. And then we can read the description for Entoloma rhodopolium group, which all of this stuff lines up perfectly. Habitat scattered or in groups or clusters on the ground in the woods, mainly under hardwoods, widely distributed. If I were to read all that to you, you'd get bored, but it really does describe this mushroom. Edibility, poisonous, causing vomiting, diarrhea, abdominal cramps that may require hospitalization. All fleshy entolomas should be strictly avoided, although some are said to be edible. They're difficult to differentiate and not well known. So there we go, Entoloma rhodopolium group, and that's what it looks like to run through an entire dichotomous key to bring us where we are. This book may be 30-something years old, but it's still super relevant, and mushroom identifiers use this book still, and it was very effective and brought us right to Entoloma rhodopolium group. So now that I have a pretty good idea of what mushroom this is, I'm going to put these into the microscope. We're going to have a look at some of the microscopic features. First, we're going to have a look at these spores, and then we're going to take a look at some of the actual fruiting body to look at the cellular structures and get some pictures of the microscopy. Now, you don't need a microscope to identify mushrooms, but it sure does help, and it's a lot of fun. If you have an interest in mycology or mushroom identification, get yourself a cheap microscope. The one that I'm using is only a couple $100 on Amazon. I'll put a link in the description for that one, and it's perfect for beginner amateur mycologists just to see some of these fungal structures like spores and the cellular structures of the tissue. So now we're going to get into the microscope part. All right, so now it's time to take this spore print and we're going to put it into the microscope. So here I have this microscope slide. You can see that spore print, I hope. Very salmon colored, very salmon pink. And for spores to make that thick of a deposit, that is a lot of spores. So one thing I'm going to do is I'm going to clean it up just a little bit. I'm going to wipe some of these spores away, except for where I'm going to put my cover slip. So just using a Q-tip, I'm just going to clear them off a little bit so my slip will sit down tight on there. Alright, and I have a little cover slip here as a very thin little piece of glass. So what I'm going to do is just a simple water mount. So I have some H2O here. You can get one of these big variety packs on Amazon for pretty cheap. And so what I do to get an accurate couple of drops is I just put a little bit of water down on my table, and I'm going to carry a couple of drops of water over here and just let it sit for a second so that those spores can fully hydrate, and then we're going to put our cover slip on. You can see all those spores right there within the water driplet. So now I'm going to take my cover slip. You don't want to get your finger grease all over the center of it. And I'm basically going to use this tweezer to kind of spread out this water a little bit. But I'm going to, yeah, we got a lot of spores in that water. Nice. Now I'm going to take my cover slip. Hard to do while I'm filming. Just going to set it down on that water. Just like that. And I'm going to just do that. And then we're going to absorb a lot of this excess water away from around the edges. Pretty clean looking slide. You really can't even see anything on there, right? I'm going to put these into the scope and we'll have a look at these spores, see what shape they are, and then measure them by taking a photograph of the spores, running it through this process, this program called Pixie Meter that measures the spores, and then we can, compare the shape and the size of the spores against what we already think we know about the mushroom to kind of really confirm its identity. All right, so I have all this mushroom material, and you could send in a piece that big to have it sequenced. It really doesn't take much, and I usually will keep some of the specimens for myself in my little herbarium. And I collect mushrooms because a lot of these I send in for sequencing, and I kind of would like to have evidence of it, a little piece of that mushroom myself. So here's an example of my little herbarium. It can be as simple as this. Look at Morchella norvegiensis, Morchella conica, or sextillata, or eximia. This one, Morchella tomentosa. So a lot of different morels, but there is just tons of mushrooms stuffed into here. So you don't need a lot of room to keep an herbarium. And on these I have the iNaturalist number written down so I can refer back to the photos, exactly where these mushrooms were found. And so it's really a cool system. So here I'm going to take my microscope and turn the binoculars around. This really isn't a lesson in microscopy, but I just want to show you what these spores look like. So I take my slide, whoops, put it right there. I get it lined up under there, and I start with the smallest objective. And yeah, I can see tons of tiny little spores here. All right, so you see all those little tiny spots. Those are spores. We can't tell much here because we're only at 40 times the magnification. So next we're going to step up to 100 times magnification and see what we see there. Focus this a little better. There we go. Now we're starting to see something. There we go. Now I can actually kind of start to make out little shapes, but we're going to step it up to the 40 times objective. So when I have these eye pieces, these are times 10, and this one is this one's times 40. So that's going to make this 400 times the size of the spore. Look at that. It needs a little bit of fine focusing. Now we can see them. Interesting spores. They are angular, so they fit the description of Entoloma. Sorry for the shoddy camera work, but I'm about to step it up to the 10 times and the 100 times, so the 1,000 times objective, which takes a little droplet of oil. So I got to put a little drip of oil there for this objective. Actually sits in the oil, and then we can see these spores at 1,000 times magnification. And again, you really can't see much going on here with the naked eye, but as you've seen, there is a ton of spores here. So let's put our oil droplet on and have a look. All right, so this is immersion oil, and I'm just gonna put one nice drop right on the top of the slide, and I actually back the stage down just a little bit so it doesn't move my cover slip, and then I slide that objective right into the oil, and now I'm gonna have to fine focus it to see where we are. There we go, angular spores. So a lot of spores are shaped kind of like a pumpkin seed. These ones a little bit more like a stop sign. So I'll give you a look at these. So there you are at 1,000 times. There's your angular spores. That matches with the description of Entoloma. And so now I'm going to take a couple photos and upload them into my computer, and we're going to run them into Pixiemeter, measure these spores, and then see if it matches with the Entoloma rhodopolium group. So yeah, let's zoom in. You can see those spores angular, so they're shaped kind of like stop signs or something like that. But I'm going to take some nice photographs, and I have the program set up to the settings on the phone that I'm actually using right now to show you this, so I can't show you me taking the photos, but just trust me, it's just like this, but just still photos. So after putting these spores into a computer program called Pixiemeter, which I'm not going to do a whole video about right now, it takes an average of all of the different sizes of the spores. So I pull this little cursor that stretches width-wise and lengthwise of the spore. I did about 23 different spores in this example, and it looks like my average is 10.1 micrometers by 8.7 micrometers. So that is our spore size, which correlates pretty well with the Entoloma rhodopolium group. But after doing a little more reading, it turns out that the rhodopolium group is actually about 5 or 6 different species just here in Washington with different spore sizes that we're calling the same thing. So kind of confusing, but the spores confirm Entoloma rhodopolium group. The microscope is super fascinating. You can go way down a rabbit hole looking in the microscope, but I have a measurement of all of these spores now. I'm convinced that I know that this is an entoloma, a toxic entoloma. But there is one other thing, and I waited to the end of this video to bring this up because a lot of people want to use this first before doing any of the work. And using pictorial keys and dichotomous keys to identify mushrooms has been the way that we've been doing it for so long. But thanks to the advent of the internet, and especially with AI now, there is a lot of useful applications out there that you can just get on your smartphone. And we already talked about one, iNaturalist, that I mentioned earlier in the video. This one has a feature where it will ask you, what did you see? And you click on that and it will give you suggestions based on AI information in their database. And it's actually pretty accurate. It can at least get you in the ballpark. So these particular mushrooms, when I looked them up on iNaturalist, it has quite a few different suggestions all over the place. So really hard to use this as a definitive key to identification. I wouldn't trust one of these apps to just find a wild mushroom, show it to the app, and then go ahead and eat it. I would definitely suggest doing my homework and discovering through literature and stuff like that what kind of mushroom it is. There's a whole bunch of different apps just like this. I suggest using the iNaturalist one just because that's the one I use. Google Lens is getting better and a lot of these are pretty accurate, but again, I wouldn't base my life on trusting AI quite yet. So use these in conjunction with pictorial keys to help get you to an understanding of the species that you're looking at. And one of the biggest things that I have found useful to identification of mushrooms and learning about mycology is to use social media platforms. Maybe you are not on Facebook because you just don't believe in it or you don't like it or whatever, but this might be a good reason to get on Facebook, is because it is chock full of mushroom identification groups, and I have learned so much through just following these mushroom identification groups on Facebook. So I hope that you found this video useful. Like I said at the beginning, we could go way down the rabbit hole. There are so many different kinds of mushrooms, but if you go into the woods and you find a strange mushroom, do the things I said. Number one, take nice pictures of it. Number two, take a specimen home with you. Number three, take a spore print. Then when you get the color of those spores, number four, I want you to get into a dichotomous key and start going through the keys. And you can use these other books. A lot of people just want to look at the pretty pictures and see ones that look close, so you can use the very limited key that's within some of these books and then just look through the pictures to find ones that look close to yours. You can use AI powered apps and you can use identification forums on social media. Another great way to get your mushrooms identified or to get a second opinion is to join your local mycological society, take your specimen into the meeting, and there's usually a bunch of people there eager to help you to identify your mushrooms. So I'll often use a lot of these different things in conjunction with each other. I'll use the AI powered app like iNaturalist. I'll also look in the books, and then sometimes I'll even run it by other people, whether it be online, just sending pictures through a text or a Facebook message, or taking them into the Mushroom Club and having other old timers or more experienced people looking at the mushrooms and confirming what I have found before I'm ready to throw it in the pan and cook it and eat it. If you follow these suggestions, I think that you can safely and confidently be eating wild mushrooms in no time fast. So thanks for joining this episode of Mushroom Wonderland. We'll see you on the next one. Much love, everyone.